कहाँ, घर पे हो?
नहीं नहीं सर, ये मैं अपने आईटी डिपार्टमेंट में बैठा हूँ।
सॉरी। मैंने अपनी स्क्रीन शेयर की है संत,
जी
आप देखिए, इज इट
सर,
विजिबल?
इट इज़ विजिबल, यस सर, इट इज़ विजिबल।
अभी मैंने फुल स्क्रीन नहीं किया है
जी
इसको।
जी सर, जी सर, इट इज़ इन, इट इज़ विजिबल, क्वाइट विजिबल सर।
अब पूरा पूरा स्क्रीन विजिबल है प्रॉपरली?
अभी सर स्लाइड शो में नहीं आया है।
अब फुल स्क्रीन पे नहीं आया था क्या?
नहीं 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 सर, अभी नहीं आया था सर। Associate Professor, ISF College of Pharmacy, Moga, would like to welcome Dr. Gaurav Kaithwas, sir, Professor and Associate Dean, Academic Affairs, Head Department of Pharmaceutical Sciences and Dean, School of Pharmaceutical Sciences, Babasaheb Bhimrao Ambedkar Central University, Lucknow, at the online platform of ISF College of Pharmacy for expert talk on as a new era in cancer prevention. Welcome, Professor Gaurav, sir. Enlighten about the purpose of ISFCP 101 dialogue series. Over to director, sir.
So, thank you, Dr. Sant Kumar Verma ji. First of all, I am thankful to our ISF Dialogue series for inviting me and provide this opportunity. Being a director of institute, this is our moral duty to welcome today's expert, Professor Gaurav ji, among us. He is a very dynamic, dedicated, and master in his the field. So, I welcome you, sir, from core of my heart, and this is the ISF dialogue series. So we want to produce some of the specific talk for the audience, researcher, young scientist, as well as of also teaching faculty. So this is the opportunity to ISF dialogue series. Professor Gaurav ji with us. So I again welcome you and request to Dr. Sant Kumar Barmaji, please proceed.
Thanks, respected director, sir, for enlightening about our 101 ISFCP dialogue series. Now, I would like to request our today's speaker, Professor Gaurav Kaithwas, sir. Over to
I couldn't hear you, Sant. I think I anticipate that you might have handed over the things to me. Am I audible, Dr. Sant?
yes, 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 sir, yes, sir, yes, yes, sir,
Okay.
yes, sir, yes,
First
sir.
of
You
all,
are audible, sir.
before I'll get into the business, I must thank Professor Gupta and Dr. Sant Kumar Verma for considering me for this lecture series. And without taking much of a time, because Dr. Sant has limited me in the duration of 30 minutes, so I would straight away get back into the business, and we will be discussing about today. We'll be discussing about the small interfering RNAs, which has come up, or which is coming up rather, as a new era in a cancer prevention. So before we get into the small interfering RNAs or RNA interference, let us try to understand what is cancer. With a very brief description, I will take up the cancer, which further turns into the tumor. The first thing is a normal cell. Normal cell are those cells which we usually see, which we usually are in our body. They are in a direct supervision and control of growth, proliferation, and division through various signal transduction mechanisms. And whenever, like our body, like a human being, or any living organism, whenever there is a cell, it will grow, it will divide, and at the end of the day, at some point of a time, it will die. And that will happen with the help of various processes like necrosis, which happens due to injury, apoptosis, which is a programmed cell death. Autophagy is also a programmed cell death, prognosis, and there are several other mechanisms of the cell death. Now, what is tumor? Tumor is a stage wherein a cell develop autonomous mechanism for its growth and proliferation. That means that the cell will not be able to control its growth and proliferation with the normally devised mechanisms. And why a cell becomes a tumor cell? Because of the carcinogens. There are several substances, chemical agents, UV rays, uh, several other agents which can induce these, or which can disturb these normal signal transduction mechanisms into the defective ones, leading for the conversion of a normal cell into a tumor cell. Overall, tumor is an uncontrolled growth of a group of cells, disregard of their normal rule of cell division. This is how a normal tumor cell formulates that, like, because, like, suppose if these are the normal cells, because of any mechanism or any carcinogen, any rays or smoking or any xyz things, they can induce, if they can disturb the normal signal transduction mechanisms of the cells for their growth, these cells, they become tumorous. They start proliferating by their own without following the normal path, and thus these cells are referred as the tumor cells. The tumor cells could be of two types. It could be a benign type or a malignant type. The benign type tumors are those tumors which usually remain confined to their original space. They are located, they don't move outside here and there. However, when these benign tumors, they start growing more once the localized space is not sufficient for them to get the energy, get the oxygen, get the glucose, then they try to malignate. They try to move out of their boundaries and reach out to the nearby tissues or to the circulatory system or to the lymphatic system. And this is how these benign tumors, they spread towards the other parts of the body, and these type of tumors, they are termed as the malignant tumors. In a brief general etiology and pathogenesis of the cancer, if I'll take up that, there are several DNA damaging agents like chemicals, radiations, sometimes viruses also. They affect to the normal cell, which leads to a DNA damage. In a normal circumstances, the it leads to the DNA damage leading to mutation. But in normal circumstances, what happens that these mutations are repaired by our own body mechanisms. In case if these mutations are not repaired, they don't get repaired, then there are three possibilities to happen. One is the activation of the growth promoting oncogenes. There are genes which are termed as oncogenes, which actually promote the growth, and that is why they are termed as oncogenes. Then there is another possibility that the apoptotic regulating genes get affected. That means those genes who are actually regulating the programmed cell death within the cell, they are altered, and subsequently the apoptotic mechanisms within the cells is affected. Then there are another set of genes which are called as a tumor suppressor genes or cancer suppressor genes. They are also available in the body whenever there is a signal for the uh, uh, promptness of the tumor cells or a cancer signal. Then these tumor suppressor genes or cancer suppressor genes, they subside these signals. In case of failure of these, any of the three mechanisms or all of the three mechanisms, this leads to the formation of a tumor and thereby malignant tumor cells. Although it is not very much relevant to the RNAi, but I thought it of not to removing of this slide. I will just briefly uh, describe this slide. Like in a normal cell, what happens that whenever we take any sort of a food, any energy, take any sort of a food to get energy, it get converted into the glucose, and through glucose, the glycolysis converts. The glycolysis happens and produces the two ATP molecules, and subsequently the end product of the glycolysis, it goes to the pyruvate. The end product of the glycolysis is the pyruvate, which further goes into the citric acid cycle and oxidative phosphorylation to produce 34 ATP molecules. Now, what happens? Why, and how these cancerous cells are so difficult to handle, or why they are so dangerous, if I say. The reason is very clear, and clear that whenever there is a cancerous cells, a tumor cell, a mass develops, and in the core of that mass there is lack of oxygen. Now, if a tumor mass is there, the core of that mass is oxygen deficient. Subsequently, what will happen that the cell which the pyruvate which has to go to the citric acid cycle or and to the oxidative phosphorylation will not be able to complete it, because this process needs very heavily needs oxygen. And subsequently, what happens, there is a continuous glycolysis which goes on, and the pyruvate, which is an end product of the glycolysis, it is not able to move to the TCA cycle and oxidative phosphorylation. Subsequently, in cancer cells, what happens that these cells, they repeatedly perform the glycolysis, and this is the most dangerous part actually, for the most difficult part of the cancer cells to handle, because once there is a repeated glycolysis in these cells, this pyruvate get converted into the lactate, and subsequently this lactate moves out of these cancerous cells or the tumor mass to the nearby cells, which are actually normal cells, normal epithelial cells. Okay? But because of the presence of the high lactate, the signal also promotes them to become the malignant. The first signal for the malignancy starts from these lactate movement across the cells, from the hypoxic cells, which are at the core of the cancer cells, then to Oxford cells, and then to epithelial cells. So this is how the problem lies with this mechanism over there. Now, a brief about the type of cancers: that the carcinomas, the sarcomas, lymphomas, and melanomas. The carcinomas are those which arises from the epithelial cells or the ectodermal cells, whereas the sarcomas, they are the part of the connective tissues and muscular tissues. For example, the bone tumors, muscle tumors. On the other side, the lymphomas or leukemias, they are also terms, which is the growth of malignant growth of the WBCs, which leads to the excessive production of leukocytes, and in routine it is termed as a blood cancer. Melanoma is the cancer which refers to the skin cancer, which develops in the cells of the skins, which are melanocytes, that produces the melanin. A brief about the etiological agents that causes or induces cancer. That could be the physical agents, which means the UV rays, ionizing radiations, biological substances or biological materials like bacteria, viruses, parasites, and the chemicals like alcohol, arsenic, and few components of the tobacco smoke. Now, these are the options which are available for the treatment of the cancer, that is, hormonal therapies, which are particularly available for the cancers like breast, okay, prostate. Then surgery is available. Bone marrow transplantation is one of the available options for the chemotherapy. Chemotherapy itself, targeted therapy, radiation therapy, and immunotherapy are few of the options available for the cancer treatment. However, majority of these options, they offer a major disadvantage, or they suffer disadvantages because of the two reasons. The first reason stands out to be that the diagnosis of the metastatic tumor or angiogenic tumor is very late, and mostly once the patient reaches to the hospitals, okay, the tumor has already gone malignant, and therefore the surgery and other therapies are not very much effective. Chemotherapy works to some extent, but the disadvantages and the adverse effects and the side effects associated with the chemotherapy are so large that the suffering to the patient goes beyond control. Now I will come to my basic topic, that is the RNA interference. So before I brief the RNA interference, I'll take up a review of the RNA structure as well. The RNA is a polymeric molecule essential in various biological roles for coding, decoding, regulation, and expression of genes. RNA and DNA are nucleic acids, and they are one of the four major macromolecules essential for the life. DNA is, as we have already studied a lot, that DNA is a double helical structure, whereas RNA is a single strand folded onto itself, and it is not at all a DNA like double helical structure. Most of the RNAs which we have studied till now might have studied all other also, but just what we have studied during our grads and schooling, that which are biologically active RNAs are the mRNA, tRNA, rRNA, sRNA, and apart from this, there are some other non-coding RNAs which contain self-complementary sequences and allows the part of RNA to fold and pair with itself to form a double helicals. So, see, most of the biologically active RNAs are the messenger RNA, tRNA, ribosomal RNA, and there are some other RNAs also which are non-coding RNAs. But these non-coding RNAs are very important, and from these non-coding RNAs, the RNA interference has emerged. I'll explain it in the next slide itself. Each RNA contains a ribose sugar, which is numbered through the 5 to 3 prime, and there are 4 bases attached to it. That is adenine, cytosine, guanine, and uracil. Now here is the type of the RNAs which I was talking in the previous slide. The RNAs are usually, they are coding RNAs. They act as a genetic materials like the DNA acts, and very rarely they are also double-stranded also. Okay. There are some non-genetic RNAs which don't at all act as a genetic material. They don't have any genetic code for any genetic message, and these RNAs are usually single-stranded. The non-genetic RNAs are the messenger RNA and the transfer RNA and the ribosomal RNAs. The ribosomal RNAs are the integral part of the ribosomes, and these are the type of RNAs which actually are the key players in decoding these RNA interference. Like the major classes of these ribosomal RNAs are the small nuclear RNA, microRNA, siRNA, heterogeneous RNA, uh, which actually participate in the uh, small RNA interferes. In this, the siRNA is the major player in these in the RNAi. Now, this how the RNAs, like how these small interfering RNAs, they actually are able to do the gene silencing, or what is the mechanism and how they behave actually while doing this. The first thing is to be understood that it was very late when it was discovered that there are small interfering RNAs in the physiological system. And why it was discovered so late? Because the half life, the shelf life of these small interfering RNAs is very small. So they live there for a very small period of time, and that is why their discovery was very late. They were discovered very late, and when they were discovered, their therapeutic potentials were exploited. So RNA molecules inhibit gene expression by disruption of the specific mRNA molecules. So what we do in the RNA interference, that we take these small interfering RNAs and we target them to a particular mRNA, so that the destruction of the mRNA can be executed. Consequently, the message which the mRNA has received from its gene will not be transformed into the protein, and that the whole process is termed as the RNA interference and gene silencing. Historically, it is also known as a co-suppression, post-transcriptional gene silencing, quelling as well. During RNAi, the double-stranded RNA is cut down into the small RNAs, which are actually three major types, which is the siRNA, shRNA, and miRNA. And these three major types of small interfering RNAs, they actually induce the RNA interferes by the help of an enzyme which is called as a dicer, which I'll be explaining in the later slides. These three siRNAs, the RNAs which are siRNA is basically a double-stranded RNA with 21 to 25 nucleotides. Okay, the two nucleotide overhangs that activates the RNAi. The same thing implies with the miRNA also. It is a single strand RNA, which is 19 to 25 nucleotide sequence, and it is encoded by a non-protein coding genome. The another type of the RNA is the shRNA, which is a small hairpin-like RNA. It is also a double-stranded RNA, which is made up of 15 to 20 nucleotides. Basically, it contains a loop structure that is processed to siRNA. So basically, the shRNA is converted into siRNA by the battery of enzyme. The whole process of RNA interference is executed by any of these three RNAs, along with the dicer enzyme, and which constitute a complex which is called as a RISC complex, the RNA induced silencing complex. That is why it is termed as a RISC. The RISC complex is basically a multi-protein complex made up of a 500 kilodalton that incorporates one strand of a small interfering RNA, which could be a siRNA, shRNA converted into siRNA or a microRNA. The RISC triggers the mRNA degradation in response to siRNA unwinding of double-strand siRNA by ATP-dependent helicases. I'll explain the process. This is a typical structure of a small interfering RNA, siRNA. Uh, as I have told in the previous slides, it is usually 19 to 20 to 25 fragments of the nucleotides. It's a double-stranded RNA, wherein the three end overhangs over the five end, and this is the overhanging end which requires it for the activation of the RNAi. Uh, if I'll take up a brief about the mechanism of siRNA interference produced by the siRNA, it includes that the double-stranded RNA is chopped into the short interfering RNAs, siRNAs, by the enzyme which is called as a dicer. Then this dicer chops it down. Then there is a three end overhang. This is the five end. Okay. Then dicer is a family of enzyme which is endoribonuclease dicer, or also termed as a helicase. It belongs to the RNase 3 family enzyme, and it cleaves the double-stranded RNA and pre-microRNA into short double-stranded RNA fragments called as a siRNA or miRNA. The siRNA dicer complex recruits additional components to form a RNA-induced silencing complex, which is called as a RISC complex. This siRNA and the dicer, they recruit several other proteins to form a RISC complex, and this activation of the RISC complex leads to the unwinding of the siRNA as well. The unwound siRNA base pairs with the complementary mRNA. Now, once this siRNA is unwound, then what happens that this siRNA RISC complex, it interacts with the mRNA, and thus the guiding RNA machinery to target mRNA. Subsequently, this three end overhangs, it interacts with the mRNA and guide it to pair with the mRNA, and subsequently leading to the degradation of the mRNA, and this finally leads to the inhibition of the protein synthesis. Now, if I'll talk about the second type of the RNAi, which is introduced or which is induced by the microRNAs. MicroRNAs are also as I have told, that these are small non-coding RNA molecules, approximately of about 22 nucleotides in size. They are found in plants, animals, and in some viruses as well. The RNA silencing and the post-transcriptional regulation of gene expression is regulated by these miRNAs. They basically function via base pairing with the complementary sequence of the mRNA molecule. As a result, these mRNA molecules are silenced and not involved in any further protein synthesis. So what is happening over here, that there is a cellular gene. From this, there is a primary miRNA is formed. Then the drosha is a gene which, is an enzyme which actually cleaves it and leads to the formation of pre-miRNA, and this pre-miRNA is further cleaved with the help of the dicer endonucleases, which leads to the formation of mature miRNA, which is actually a single strand miRNA. This miRNA then further recruits the mRNA and leads to the degradation of the mRNA, leading to the translational inhibition. Explained it. I'll explain it in a more elaborative way, that the pre-miRNA is the primary mRNA that are transcribed from the genome, that are first cleaved by the nucleus by the drosha, and once the drosha cleaves it, which is also RNase 3 endonucleases, this fragment is usually around of 70 nucleotides. This is called as a pre-miRNA, primary. That is why it is called as a pre-miRNA. And subsequently, that there is this is a precursor. That is why it is called as a pre-miRNA, and it reaches to the cytoplasm through the channel which is called as a exportin 5, where it is further cleaved by the dicer, which is again a RNase 3 endonucleases, and which produces mature miRNA segment. The mature miRNA segment is generally double-stranded, and it further loops back into the form of a hairpin-like structure, which is actually a shRNA. This mature miRNA then further incorporates in the RISC complex as it was going on with the siRNA complex, and this leads to the cleavage and degradation of the specific target mRNAs. Then the third type of the RNA interference is produced by the short hairpin like RNAs, they are intermediate products of the either of the siRNA or of the miRNA. These are termed as a short hairpin RNAs. As we can see through their structure, they look like a hairpin and are made up of a 20 to 15 to 20 nucleotides. And because they have a loop structure, they are called as a hairpin RNAs. And they can lead to the degradation of mRNA in a sequence-specific manner, depending upon the complementary binding of the target mRNAs. The expression of shRNA in cells is typically accomplished by delivery of plasmids or through viral or bacterial vectors. So now these shRNAs, short hairpin RNAs, they can be delivered. They can be actually delivered into the cell with the help of various vectors. Sometimes maybe we can use plasmids. Sometimes maybe we use viruses. Bacterial vectors are also available for the delivery of these hairpin RNAs or the siRNAs also. In addition to these plasmids and vectors, we can also use the lipids, sometimes nanolipids, formulation based products. Means we can actually incorporate these small interfering RNAs into the lipids so that they can be delivered into the cell. The other way out for delivering these small interfering RNAs is the electroporation. With the help of electric fields, they can also be delivered into the cell. Uh, the shRNA is a slightly advantageous mediator of RNAs as it has a very relatively low rate of degradation and turnover. The major challenge, I would like to mention that the major challenge over here with the RNA is that these small interfering RNAs, they are very short-lived, as I have told earlier. So it is very difficult to sustain them in the physiological system for a long period of a time. So we need to deliver them with the help of one or other material which actually helps them to hold for a longer period of a time in a systemic circulation, and decode them or release them once they reaches to the respective site where the action is sought. The another problem with these RNA interference molecules is that they have the off-target problems, that they can target to the other mRNAs also, which may lead to the adverse effects. Although there are several mechanisms and methods through which we can actually remove these off targets, and various new methods and mechanisms have been developed for the delivery of these small interfering RNAs into the systemic circulation. The shRNA offers further advantage that it has a long-lasting gene silencing effect and is of a great interest for gene therapy. Both siRNA and shRNA use common molecules in their pathway, including dicer RISC complex, and their aim is for the degradation of MF. So this is the basic mechanism for the shRNA interference, as I have said, or I have explained with the siRNA. The mechanism behind both the shRNA and siRNA is typically same. They utilizes the same dicer enzyme along with the RISC complex for the degradation of the mRNA. Now, if we talk about the utility of the RNA interference in cancer, the different type of activated oncogene mRNAs can be effectively inhibited by RNA technology to inhibit tumor growth. Tumor has been decoded as a multiple a disease with the multiple factors involved in it, and that is one of the reason for controlling the tumor growth once, particularly when it has gone into the metastasis and angiogenesis. However, RNAi, since it targets a specific mRNA or it targets a specific gene, so to be get silenced, so it has very high efficacy and potential through the induction of silencing even at the advanced stage of growth, even at the metastasis and angiogenetic stage. That means we can specifically target the angiogenetic and metastatic genes, so the further growth and could be stopped, and subsequently the chemotherapy would behave more effectively, and that is why they are moreover they are highly specific methods for the cancer therapy in comparison to chemotherapy. They have a precise functional mechanism. They can target multiple genes of various cellular pathways involved in the tumor progression, and that is why they are more effective and highly efficacious also. uh, one we have been in we are living in the era of personalized medicine, and RNAi could be a therapy for a personalized medicine, because we can actually study the genes which are actually involved in the progression of carcinogenesis in a particular patient, and we can develop a personalized RNAi therapy for a particular patient. And these are the advantages for the of the RNA interference in the cancer chemotherapy, whereas the cancer therapy. Whereas if we talk about the applications, they can be used for the inhibition of tumor anti-apoptotic genes. They can be used to study the tumor signal transduction pathways, inhibit the tumor angiogenesis related factors. They can affect the tumor oncogenes and the tumor suppressive genes, and also reduce the tumor drug resistance. Uh, Despite having all the benefits, despite having all the advantages of the RNA interference, it has several barriers which needs to be overcome, which needs to be worked upon. Like the naked siRNAs are unstable in serum, and they are very easily degraded by the nucleases. That's why they have a very short life. The siRNAs are engulfed by the reticuloendothelial system, which is composed of phagocytes, which is one more way of contributing to the short life of these siRNAs. Since the size of these RNAs is very small, they are also susceptible for the renal filtrations, and they get very easily filtered by the kidney. So overall, all these three factors, they contribute towards the short half-life or short life of these small interfering RNAs in the physiological system, and the people are working out towards overcoming these uh, barriers. The other type of problems with the siRNA or RNA interference is that the tumor capillaries are discontinuous and have large openings, which are conducive for the transmembrane transport of siRNAs in tumor tissues. So that means they suffer the transmembrane transport within the tumor tissues, because sometimes they have a large opening, or sometimes they have a very small openings. So this also needs to be worked out. Apart from these, the cell membrane is negatively charged, and siRNAs are also negatively charged and water soluble, and that is why we need some mechanism for these siRNAs to pass into the cells. As I have told that we can do the electroporation, we can use some bacterial vectors or viruses, or sometimes of a lipids like lipofectamine can be used to overcome this cell membrane barrier. Moreover, once the siRNA reaches to the target cell, it is encapsulated in an early endosome for transport and fusion with the late endosome. The late endosome causes the pH to become acidic and fuses with the lysosome to transport. At very low pH, which is around 4.5 to 5, and due to nucleases, the RNA molecules in the delivery system are degraded. So that means not only in the physiological periphery, we have to secure their delivery within the cytoplasm also. We have to secure their delivery because of the low pH and nucleases. Uh, One problem is a big problem with the RNA interference is there is off-target effects. It's very simple. If I simplify it in a normal pharmacological way, that whatsoever the drug we take, the drug has some sort of a side effects, and the same thing is offered with the RNA interference also. Whenever we have designed, or when we have prepared a small interfering RNA, it may have a matching complementary sequence with some other mRNA also. Consequently, this may lead to the degradation of the other mRNA molecules, which may lead into the several other uh, degradation or non-responsiveness of the gene response, ठीक है, because of the degradation of the mRNA of several other proteins. So this is one of the another. This is one of the other challenges which lies with the RNA interference. This is a small team. Thank you very much for your listening. That's end from my
Thank you,
side.
sir. Yes.
Shall I
Thank you,
close?
sir. Sir, I would like to invite director, sir, for thanks to our speaker.
Thank you very much, sir, and very nice, informative, scientific talk. So we all are the thankful to you, sir, for accepting our invitation and provide a great and scientific information. And this lecture it will be the uploaded on our YouTube channel. So we have the more than 5,000 of the subscriber of our own channel as well as our number of the students. They can connect with you, and I also request you, as and when you are in this side in the Punjab, you're welcome to visit this institute and give your blessing as well as the your suggestion, so that we will grow the at a sharp level. As you know that in the private system, ISF college team is doing the number of the good job, but sometimes we have some of the limitation of the fund. So I also expect from your side, if you feel that the ISF is the suitable and comfortable, then we can also do one of the MOU with you, because I have the MOU with the NIPER institution, and we have recently forwarded the application to the Central University Batenda, and Vice Chancellor appreciated the efforts taken by the ISF College of Pharmacy. Sir, we are running one of the NABL accredited lab inside the institute, and some of the big institute like the LCMS and X-ray diffraction. So such SAM, TAM, this types of the instrument we have not, and this types of the facility at your end, so that we can also do some of the joint work. So again, I am thankful to the core team of the ISF Dialogue Series, and this dialogue series we are the running with the IIQ, our team as well as the IIC. So IQAC coordinator Dr. Siddharth Manji and IIC coordinator Dr. Pooja Chawla both are taking lot of the pain for strengthen the activity, and thankful to the Dr. Sant Kumar Verma for invite Dr. Gaurav ji and interact. This is a platform. So even not we are in the offline mode, but the online mode, this types of the things create the lot of efforts and their queries. I will discuss with the expert, and then we will reply to you in time. So thank you very much for accepting our invitation. Thank you, sir.
Thank you very much.
Thank you, sir. Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir.
May I leave?
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes.